Hello, welcome back. This is Professor Young, and this is part one of three of our look at the skeletal system. I've decided to chunk this a little bit smaller just because of the detail and the wide range of information that we have to cover for the skeletal system. This is really at least what I would consider to be our first look at a system. We looked at the integumentary system, but as far as the major systems go, I kind of consider this our first real organ system that we are considering and looking at. And so with that said, why don't we go ahead and jump in and get ourselves started. As always, as we go through this, remember if you need to rewind or fast forward or skip ahead, please feel free to do so. And also, if at any point in time you have a question, please jot it down. And then as soon as the WIMBA session is over, either go back over and re-listen to that section, or feel free to shoot me an email with that question so that way you can get the answer before you forget what the question even was. So I guess the best place to start is to try to identify exactly what good bones are in the first place. And to do that, why don't we start with the obvious, and that is bones are good for support and movement. If we didn't have that skeletal framework in place, we would be a blob, just very fluidly moving around the floor. You wouldn't be able to sit up. You wouldn't be able to sit or stand up. You wouldn't be able to walk. You would just be this amorphous blob of skin and organs kind of gliding along the floor. And that's not much fun. And so support, structure, that's an obvious given as far as what bones are good for. But additionally, bones also aid in movement through the attachment of muscles. Muscles are a great and wonderful organ within our body. Unfortunately, without something for them to move and without something for them to attach to, they don't really do much good. And so muscles need to be able to attach onto something that they can physically then move. That attachment is bone. And we know that muscles attach to bone via ligaments. All right, muscles attached to bone via ligaments. And this is something that you will end up seeing. Oh, not ligaments. I'm sorry, I lied. Scratch that. Don't pay any attention to the man talking into the microphone. They form attachments through tendons. Sorry about that. Remember, bone to bone is a ligament. Muscle to bone is going to be a tendon, and this is going to be something that you will be able to see when we get into the dissections. Now, how exactly do bone or does bone function? This may be a set of functions that you're not aware of. I think everyone gets the fact of support and muscle attachment. But bones do a lot more than just that. There is more of a greater function for them. First and foremost, it is a reservoir or a storage area for calcium. And specifically, both calcium carbonate and calcium phosphate. Right now, these are both called biosalts, or calcium salts, if you would. This is a salt precipitate. We know that bone, being connective tissue, is composed of the three basic components of all connective tissues. We know that they have fibers. We know that they have cells, and we know that they're made up of amorphous ground substance. We know that the main cell type within bone is an osteocyte, although there's some other derivatives in there that we've talked about. The fiber type we haven't really talked about yet, but we will very briefly. I mean, it's basically collagen fibers. And then there's this amorphous ground substance. And remember we said, amorphous ground substance can range from anywhere from the fluidity of plasma in blood to the firmness found within bone. And what allows that amorphous ground substance to become so hardened is this presence of calcium-based salts that get absorbed back into the bone structure and ossifies or solidifies or hardens that amorphous ground substance matrix. We're going to talk about exactly what controls that towards the end of this WIMBA session. Right? But for right now, know that calcium is stored within the bone, and it's stored there for a multitude of reasons. One is the regulation of the blood calcium levels, which does tie into the next point that you see on here, which is acid-base balance. Because what can happen is bone can release calcium, and calcium can almost, as calcium carbonate, almost acts as a buffer to try to restore normalcy if blood becomes too acidic. And so that's one major reason why we need a reservoir of calcium. Secondly, as we will find out in lecture very shortly, muscle contraction requires a lot of calcium. And so, since bone and muscle are so intertwined with one another, it kind of makes sense for there to be a reservoir of calcium stored right nearby where the muscle might need it. And as we will also find out a little bit later down the road, that when we talk about neural conductivity, or the way that the nervous system sends messages down one neuron to the next neuron to the next neuron to the effector, or wherever it's going to innervate, that's also going to require some calcium as well. And so the body relies on that calcium for quite a bit, much more than just hardening the bones. We talked about the acid-base balance here very briefly. Adipocytes, bone becomes a storage area for adipocytes within the long bones, within the femur, the humerus, within the ulna, the radius, within the tibia and the fibula, within those long bones, down the length of the bone, there is something referred to as the medullary shaft. We'll see it in one of the other WIMBA sessions. That shaft is made up of yellow bone marrow, and literally it has a yellowish, light yellowish tint to it, and is very greasy in appearance. Well, that bone marrow is made up of fat cells. It's made up of adipocytes. And probably the number one, in my opinion, most critical function of bone is a process known as hematopoiesis. And with hematopoiesis, what we see is the production of red blood cells. In fact, that is what hematopoiesis means. Hematopoiesis is the production of red blood cells. Hemato here meaning red blood cells. Poiesis means production of. And where we see hematopoiesis taking place are within the flat bones, such as the cranial bones, the scapula, the sternum, the ribs, the pelvic bone, and then we have the ends of the long bones, specifically within the heads and the condyles on each end. Those bones are made up of what we refer to as being spongy bone and contain red bone marrow. And it is there where red blood cells are produced. So, I think we've already been over this. I think we are pretty confident in the answers to this. All right. We know that bone is a type of mesenchymal derivative, and we realize that mesenchymal derivatives is just another fancy word for connective tissues. All right, and so what we're dealing with, I spelled connective tissues wrong. So what we're dealing here is with connective tissues. Now, we've already discussed to some extent the three parts of connective tissue. We know we have amorphous ground substance. We've talked about the fact that these calcium salts are going to cause that to harden, and we'll see that again on the next slide. We've talked a little bit about fibers, and we're going to talk a lot more about these bad boys right here, about the cell types. Right? That's something that we have yet to really discuss in detail, and we will before we get out of this session. So here they are. I've already discussed these guys. Kind of set you up for that a little bit. All right, there's your calcium phosphate and your calcium carbonate. We know that these are the culprits that lead to hardening of bone, which is a good thing. That's what we want. And within that same breath, I've also already mentioned to you the fact that collagen is the main fiber type. Now, what regulates how much calcium salts or mineral salts are in the bone? What regulates the production of the fibers? That comes down to the next slide. The cell types. And this is where really the work of the skeletal system is taking place. There are 4 basic cell types that you need to be familiar with. 3 of these cell types are related to one another. 1 of these is not. Now the question is, which 3 are related to one another, and who's the odd man out? Well, I'm going to give you the answer to that question. And the answer to that question is those 3 guys there, osteoprogenitor cells, osteoblasts, and osteocytes, are all related to one another. Osteoclasts are the odd man out. Osteoclasts, because of their function, are not related to the other 3. And really what we're dealing with here is we're dealing with cell lineages. Right? This is something that's going to be that's going to come back to haunt you a couple other times through especially AMP2, but as you study anatomy and physiology overall. Some cells have to go through a very complex pathway of development. Red blood cells, white blood cells, platelets, macrophages, and bone cells are examples of cell types that go through a process. And typically it is a differential process. In other words, one cell type kind of differentiates into another, until it becomes the final product. And along the way, each of these different or various stages of cell types have different functions and different purposes. And that's exactly what we see here with bone cells. Osteoprogenitor cells more or less serves as stem cells that are going to lead to the production of osteoblasts and osteocytes. Osteoblast cells have the job of production of bone. And so when we produce new bone, it's the osteoblasts that are doing that. Osteocytes, on the other hand, maintain bone. And so once the bone is produced, osteocytes provide integrity support, if you would. And then we have osteoclasts, which is actually part of the macrophage lineage, and they do one thing. They break down bone. And so the entire origin of osteoclasts has nothing to do with the production of osteoprogenitor cells, differentiating into osteoblasts, differentiating into osteocytes. Instead, macrophages are their own separate lineage. They have their own separate differential process that they follow. And so therefore this is the odd man out, both in lineage and in function, because the other three work directly to build and maintain and support the bone, whereas osteoclasts break it down. Right? Macrophages, macro meaning large, phage meaning to eat. And so macrophages is a lineage of cells that include osteoclasts, whose whole purpose in life is to eat large particles. They break things down and release byproducts based off of that, as we will see. All right. So real quick, osteoprogenitor cells, as I've already mentioned, are stem cells of the bone. They're going to give rise to this lineage of cells, including osteoblasts and osteocytes. Osteoblasts, once the body decides, all right, we need to make more bone, what happens from here is osteoprogenitor cells will differentiate into osteoblasts. Once the osteoblast is formed, that osteoblast can begin to lay down new bone. Now, remember back to the structure of an osteon. Within that osteon, you have the central canal. And then directly outside of that central canal, we have a ring of new bone. That is where your osteoblasts are functioning. They are functioning within the adult, right outside of that central canal, to lay down a ring of new bone. Remember, as that ring of bone is laid down, all the other rings get pushed to the outer edge and form older layers of bone, which is where we'll get to next. Osteoblasts are still functioning within the lacunae. The lacunae are still attached to one another by the canaliculi. So all of that is normal. Right? Nothing there has changed really for us. And so here's our lacunae. Inside of that lacunae is an osteoblast. And what ends up happening is this. That osteoblast begins to lay down bone. Now, I like to liken this to someone who is spending a lot of time mopping or waxing their kitchen floor. And as they're mopping and waxing their kitchen floor, they're going along and they're laying down all this wax, or they're mopping everything. And as you can see, they've accidentally started from that outer edge. And by the time they're done, they've realized, crap, I've completely sealed myself into the center of my kitchen. I have this beautifully mopped floor. It's shiny. There's not a speck of dirt anywhere. It looks brand new. And I'm stuck in the middle of it. And that's exactly what the osteoblast does. It lays down all of this new bone within the lacuna to the point where it ossifies itself right into the center, and it can't do anything else. It's stuck. Since it can't lay down any more new bone, it's completely surrounded. It has no choice but to go ahead and differentiate into the next cell type, which for us are the osteocytes. Right. And so the osteocyte differentiates from the osteoblasts. Again, the purpose of the osteocyte is to maintain bone. It's going to be laying down collagen fibers that are going to help to provide stability to that matrix of calcified bone that has been created. Think of the collagen fibers as being, if you've ever watched someone build a foundation of a house, you'll notice that when they put the cinder blocks down, all right, what they end up doing is they end up putting rebar, metal, these metal poles, down through the cinder blocks in the openings. And the reason why they do that is because when they pour the concrete down the center of those cinder blocks, it provides a structural support for that concrete. It helps to solidify everything up that much tighter. And that's kind of what the osteocytes do for that calcified bone. By laying down the collagen, it basically ties everything together that much tighter. Now, at some point in time, oh, and by the way, and these are your lacunae, and this is what you're kind of used to seeing. All right, the difference here is these lacunae are not osteoblasts. These lacunae are containing the osteocytes. And what you're looking at here, you're looking at those concentric lamellae that are extending out There's your concentric lamellae. There's your concentric lamellae. Here's another ring of concentric lamellae. Notice that the lacuna sit on them. Inside of those lacuna are going to be the osteocytes. And again, the osteocytes are going to be there to support the integrity of that calcified amorphous ground substance. Now, at some point in time along the way, something needs to trigger the osteoclasts to begin to break down bone. And a couple things happen. Osteoclasts are going to break down bone through the release of an acid. And that acid begins to basically eat away at the calcium. And as it does that, it creates an additional structure that we refer to as the pit of reabsorption. And that pit of reabsorption, and this should be a P, that B there should be a P. The pit of reabsorption is exactly what it sounds like. As the acid is released, it forms this crater within the bone. It basically forms a crater where the lacunae used to be, or that region of bone used to be, and sitting inside of it is now this dissolved calcium phosphate calcium carbonate mixture. Well, once that calcium has been released and dissolved from the bone, it is free to be reabsorbed back into the blood. And that's exactly what we see happening here. Now, I go back to my original question, and that was, what exactly regulates or controls the production and the breakdown of bone? And to do that, what I would like to do is clear everything out and kind of go to a blank slide that will hopefully give us a little bit more insight as to what is happening here. So let me start with the beginnings. Right? Let's assume that we've just had osteoclast activity, which means that calcium has been released. If calcium has been released, then our blood calcium levels have increased. So, calcium has been released, and what this means for us is that we have had now an increase in blood calcium levels. Right. Blood calcium levels are monitored by two different organs that release two different hormones. The two structures, the two organs that we are looking at here are the thyroid and the parathyroid. Right now, I think in a previous WIMBA session or lecture, I think I made mention of the fact that the thyroid there is located right beneath the larynx. And on the back side of the thyroid are these four oval glands that we refer to as the parathyroid. Well, the thyroid monitors elevation in blood calcium levels. Parathyroid monitors the decrease in blood calcium levels. Together, what they're really looking at here is, or how they're really working in tandem with one another, is as a negative feedback system. And that's exactly what we see going on here. One is going to regulate the other to a certain extent. And so as blood calcium levels increase, what we see taking place is the thyroid releases a hormone known as calcitonin. Right? So the thyroid is going to release a hormone known as calcitonin when blood calcium levels increase too high. Calcitonin is a hormone that acts specifically on the osteoprogenitor cells, that forces them to differentiate into osteoblasts. And we know that the osteoblasts are then going to create new bone, and when they create new bone, they're going to pull the calcium from within the blood back into the bone to ossify up that amorphous ground substance. And so the calcitonin is going to basically spark osteoblasts, which is going to create new bone. Now, when the osteoblasts are doing their thing, what we see is a decrease in blood calcium. We see a decrease in blood calcium. So we've got new bone being produced. We've got a decrease in blood calcium that is going on. Once the levels drop to a certain percentage, the thyroid indicates, all right, blood calcium levels are low enough, shut off the calcitonin. And that's what it does. It shuts down the calcitonin, which stops the osteoblast activity. And at that point in time, the osteoblasts go from producing bone, and they differentiate into osteocytes to be and play a role of maintaining that bone. Now, somewheres along the way, blood calcium levels continue to decrease. All right? When blood calcium levels decrease too low, what ends up happening is the parathyroid kicks in. Right? So the parathyroid is going to monitor a dramatic drop in blood calcium levels, right? Below threshold, right? Below the homeostatic level. And when that happens, the parathyroid is then going to go through and release. That's going to be a horrible color to use. It's going to release parathyroid hormone. Right, that is abbreviated PTH. The release of the parathyroid hormone directly impacts osteoclasts. And so parathyroid hormone says, whoa, or the parathyroid says, whoa, blood calcium levels are dropping too much. Something's going on here. It releases parathyroid hormone that takes the osteoclasts and forces them to break down the bone. And so as they are breaking down the bone, that's just horrible. As they are breaking down the bone, what ends up happening is we have a breakdown of bone, which increases blood calcium levels. And then, as those blood calcium levels increase, we kind of go right back to where we started at, in that when the levels become too exceedingly high, the thyroid releases calcitonin, which is going to spur on osteoblast activity, which is then going to pull the calcium from the blood, which is going to lower the blood calcium levels. And if it drops too low, the parathyroid detects that, which is going to release the parathyroid hormone, which is going to cause a breakdown of the bone by activating the osteoclast. And as you activate the osteoclast to break down the bone, you create the pit of reabsorption, which is then going to have that calcium available for reabsorption by the blood, which increases blood calcium levels. And it starts all over again. There needs to be a balance between what is happening between the osteoblast and the osteoclast activity, which is why the thyroid and the parathyroid almost work in conjunction with one another to make sure that at no point in time do we have more osteoclast activity going on and then decrease in osteoblast activity. Because think about this. If you've got all osteoclast activity and no osteoblast activity, you're breaking down bone much faster than what you're rebuilding it. And that causes weak, brittle bones. And likewise, if you've got more osteoblast activity going on than what you do osteoclast activity, that means you're building bone faster than what you're breaking it down. Then you've got a bone density issue. You've got thick, heavy bones, which is going to impact mobility. And we're going to talk a little bit more about that during the third WIMBA session for this. Right, but that raises a key issue. Think about this. Women that are of a certain age, let's say 50-ish, tend to go through that change known as menopause. One of those changes usually has an impairment of the thyroid gland. This is why women who go through menopause need hormone replacement therapy very often, because part of that regimen are the hormones that are released from the thyroid. Because the thyroid kind of burns itself out. It starts to shut down. It's also why women that go through menopause are strongly recommended to take a calcium supplement. It's also why women that start to go through menopause begin to have bone density scans done. Because what we know is, as that thyroid shuts down, we have a decrease in the levels of calcitonin, which means we have a decrease in the amount of bone production that's going on. The parathyroid's fine. Osteoclast activity is happening just wonderfully. But there's a decrease in the level of the calcitonin, which means you have a decrease in the level of bone production going on. With that said, when you break down more bone faster than what you're building it, then what you end up having is a disease known as osteoporosis, brittle bones, fragile bones. The bone density is decreasing. And so there is a lot of relativity here that I think a lot of people can probably connect to in one way, shape, form, or another. And I think this is our real first example of that interconnectedness that we have within the human body. Yes, we're dealing with the skeletal system, but while doing that, you now have an introduction into the circulatory system. You also have an introduction into the endocrine system. All right, and so there's no way to isolate out these systems and just talk about one specific system at a time. And with that, I'm going to leave you to kind of review what it is that we have talked about. And again, if you have any questions, feel free to shoot me an email or jot it down and ask me during class. And with that, happy studying, and I'll see you guys all in a bit.